Good afternoon, everyone, and welcome to today's Ecology Live, the seminar series brought to you by the Ecological Society during the lockdown period. I'm Pete Manning, and I'm chair of the events committee for the British Ecological Society. I'll be hosting today. And before we start, I just want to say a few things. Firstly, I'd like to introduce you to the British Ecological Society, if you haven't come across it before. We're the largest scientific society for ecologists in Europe, and we're very happy to be bringing you the seminar series today. There's been enormous interest in the series, and we thank you all for coming and for your continued interest in the series. Before we start, I just want to say two things. Firstly, I'd like to thank our sponsors, NHBS, who are suppliers of books and equipment for ecology, and they're offering, as part of the sponsorship deal, an offer of free shipping to European customers, and there'll be more details about that after the talk. I also just want to say a few things about questions. If you'd like to answer a question, then there's a little question and answer box. You can put in your question in there, and towards the end of the talk, we'll select a few select questions to ask Enrico. And questions which can't be addressed, there's often too many to be for all to be dealt with. We'll answer them on the YouTube version of the video, which will be posted online after the talk. So at this point, I'd like to hand over to Enrico Rezende, who is going to talk to us today about temperature effects in organisms and communities. Thank you. We just hand over.
There you go. I presume I can be seen, right?
Yes.
Hello all. Thank you so much for the opportunity to speak in here. And I hope everyone at home is healthy and safe. So let's try to start with the presentation itself. I presume you're all seeing my screen right now. And so the idea is to be talking about temperature effects in organisms and communities. And of course, that means that we're going to be talking about temperature and climate change, or global warming, if you want to be a little bit more precise. And let me see if, there you go. So before we start with the presentation, let me give you a very, very brief overview of the format that I'm talking about here. So as you can see in the slide, we're going to have some figures and graphs, but for all those at home that are interested on where the information is coming from, the references, they're shown at the bottom right corner of the slide, okay? So let's put the talk in perspective. Essentially, the main thing that we have right now is that when you look around at the world, you do have this massive spatial gradients, right? And so you have something like a latitudinal diversity gradient that essentially have more diversity towards the equator. Essentially you have higher species richness. In many instances, it's also a higher phylogenetic richness as well. And when you compare that to, for instance, what happens to marine biodiversity, you find a similar pattern. So essentially what you see in this case here, as you can see now in the left plot, is that there is some sort of association between what would be sea surface temperature, so temperature, and normalized diversity. So for some reason, it seems that you have, when you go and look at around the world, you do have at spatial scales an association between diversity, the number of species, the richness of species, and ambient temperature, right? These are spatial patterns. However, if we put things in context, we also have now the mounting pressure of global warming, right? Climate change in general, but definitely global warming is a more specific term when it comes to the rise in global temperatures. And essentially what you see here is what the predictions say. So we have the plot now of what the International Panel for Climate Change are showing. And essentially the predictions tell us that until 2100, so by the end of the 21st century, we might have something around an increase in temperatures in the order of, let's say, between 2 and 4 degrees. And as you guys can see here, essentially you have different scenarios. The uncertainty is obviously very high, but the important thing to consider is that considering the variability across models, the general consensus is yes, temperatures are increasing. By now we know this better than, let's say, when the report came out in 2014. We've seen a lot of these increasing temperature and temperature variability happening in real time. And the different scenarios here, depending on the amount of greenhouse emissions that you have might definitely affect more or less the temperature that's coming. Now, this is a temporal pattern, right? So essentially we do have an association between big latitudinal trend, a big spatial trend, but we also have this temporal trend of increasing temperatures now in time. And one of the problems that we're seeing is that when it comes to rising temperatures in time, for some reason, it seems to be associated with higher extinction rates. And so here we have now the results of one paper by Urban in Science. And what you do see is, once again, you have the different scenarios predicted here, depending now on the amount of emissions. So you have RCP 6 or RCP 8.5 that becomes higher and higher in terms of greenhouse emissions. And you have also a higher expected temperature rise. And what you see now is that with this increase in temperature, the predicted extinction is also increasing. And now you have the model here. These places actually are different predictions from different studies. So this is a meta-analysis across multiple studies, across multiple locations. And essentially what this meta-analysis shows is that the higher the impact of temperature, the higher the temperature increase, the higher the predicted extinction rates, right? So we do have on the temporal axis, this increase in temperature is associated now with higher extinction rates. Which brings us to the big question, which I'm trying to somehow address in this talk, which is how can we reconcile high diversity in warmer regions, right? When we talk about this analysis in terms of spatial scale, you do see an association between warmer temperatures and diversity. But when it comes to climate change and global warming, you seem to have this association between rising temperatures and a lower diversity. As temperatures rise, you have now a drop, or sorry, an increase in extinction rates, a drop in diversity, right? So, and this brings to the overview of the talk. So essentially, what I'm going to be talking today about is the conceptual framework that most thermobiologists are employing nowadays to try to address this question. Then I'm going to overview a little bit what the recent findings, the most recent findings on the subject employing this particular framework. And then I want to talk a little bit about the limitations of the framework that we're using right now. So essentially it's far from perfect, and I want to discuss a little bit why. And finally, I'm going to talk a little bit about the future directions, where we might be heading now in terms of thermobiology and the possibility of advancing this field and making it, you know, let's say more useful, if you will, for decision-making and conservation efforts. So let's start with the conceptual framework, right? Essentially, during, let's say, maybe the last 40 years, thermobiologists have been employing what we call the thermal performance curve, which is essentially this relationship that we have now in this left plot. And the performance curve, it shows the following relationship. It says that any given attribute, some particular trait that can be defined as performance, such as metabolic rate, it could be the rate of population increase, anything that you can actually define as a rate generally increases in an exponential manner from lower temperatures to higher temperatures. That's this increase here that you can see in the left side of the curve. And then eventually it reaches a maximum. And then we call this the optimal temperature, right, where performance is maximized. But then after this particular temperature, you see the detrimental impacts of a higher temperature. And you can see now performance decreasing until it reaches a minimum, right? Eventually it drops to zero. And so to introduce everybody to the jargon, we have the limits of this performance curve, which is called the critical thermal limits. So that's CT for critical thermal limits. And you have the CTmin, that's the limit where performance drops to zero at lower temperatures. And you have CTmax, the limit where performance drops to zero at higher temperatures. You have the thermal optimum, as I said, where performance is maximized. And you have the thermal breadth, essentially the temperature, that the breadth of temperature in which a particular organism can actually sustain performance, can remain alive, if you will, all right, depending on how we define the trait, okay? Now, the thermal performance curve has been applied to many different problems, and the idea now is to keep improving it and use it, you know, for more and more, let's say, because it has been a very useful conceptual framework to be using. And so you can see that as possible applications, you have the study of the range of thermal tolerance, right? What's the maximum temperatures and the minimum temperatures that some particular organisms can tolerate. That helps us building, ideally, mechanistic niche models, which will tell you things like the range of distribution. And also, it allows us to estimate things like the safety margins, right? So how much higher temperature these organisms could tolerate as temperatures keep rising. And so, having this performance curve in mind, we can start addressing these questions. Now, what are the advantages of the conceptual framework? I think there's one that has been, extremely useful. It's a huge advantage, which is it's a highly general framework. One of the things that I'm highlighting here is that the thermal performance curve, the typical shape that I showed, so essentially that asymmetric curve where you have an exponential increase at lower temperatures and then that sudden drop, when you look around at different traits and different organisms, you can see that it's everywhere. So for example, in this study, which I carried out with a colleague of mine last year, we're plotting here essentially photosynthetic rates in plants, so that's shown in blue. You have running speeds in lizards, which is shown in red, and you have the intrinsic rate of increase in arthropods. Most of them are insects, which is shown in yellow. All right, so you have three different traits, which we can see here. So one is at a particular level of organization. For example, fitness or intrinsic rate of increase is in yellow, it's at the population level. Running speeds in lizards, you're measuring it in single individuals, so that's something that you can measure at a particular organism. And the photosynthetic rate is actually measured at a biochemical level. In many instances, it's actually measured in leaf extracts. And so that's something that's happening at a cellular or a biochemical level. And so what you can see here in this right plot is that what we've analyzed encompasses not only many levels of organization, it also encompasses many different lineages. And you can still see the curve there, right? So in this case, the curve centered around the same optimum. So we just centered it at around the average temperature optima across this data set, which was 32, 33 degrees. And we expressed the performance in relative terms. So that means from 0 to 1 as a percentage, right? So the maximum performance, we set it to 100%. And what you can see is not only that the curve is general, but you also seem to have some general patterns, which I'll talk a little bit more later. Now, that's just to highlight the point that the performance curve is actually highly general and extremely important. And so let's talk a little bit about the recent findings associated with the application of this framework. One of the interesting things, and I think that was, let's say, a very important study, historically speaking now, is one that has used the thermal performance curves to invert the predictions of what might happen with climate change when we start understanding how organisms respond to temperature. And so to put things in perspective, what we have here in this slide is the predictions from climatologists. Essentially, which are the regions that will be most affected by global warming. And when you ask a climatologist, so when you see weather forecasts, right, climate forecasts for the future, what you can see is that some regions of the globe are going to warm up more than others. And what's happening here is that you have a higher warming up at higher latitudes. And not only that, it's going to be higher at the northern hemisphere. Okay, you can see this with this NASA in this case, it's a study. This is satellite data, but this is the reported data, but the predictions also, the models, let's say, the climate models, they also agree with the empirical data. Okay, so you do have the spatial distribution where warming is higher in the northern hemisphere and actually at higher latitudes. That would imply that these ecosystems would be more threatened by global warming. Right? You have a higher impact of temperature, and in theory, these ecosystems would be more vulnerable. They would be less resilient because the stress, let's say the perturbance is higher. But the interesting thing is, when you start looking at the animals in these environments, the thermal performance curve actually does not differ that much. And so in general, and what I can show you here in these two plots is that tropical organisms and temperate organisms, they show more or less the same higher critical limits. So that's CTmax, and you can see here, I've drawn it schematically here at 40. All right, so it's more or less the same CTmax. And when you compare now present time temperatures with that particular CTmax, so that's what you have here in blue or in red, what you can see is that organisms that live in temperate environments, so at high latitudes, the current temperatures are actually substantially farther from CTmax than what you observe when you're looking at tropical organisms. What it means is that the delta T, right, the difference between current temperatures and the thermal limits are actually higher in temperate organisms in comparison to tropical ones. And this difference is what many people are calling nowadays thermal safety margins. So that means that actually, even though warming is going to be higher at higher latitudes, the thermal safety margin of organisms inhabiting these environments is actually higher as well, okay? And when you do this sort of analysis and you generalize it, this is what you see. If you take a look here, you have now the predicted impact of climate change by 2100, so by the end of the 21st century, plotted against latitude, okay? And the two plots show the same thing just because different traits, but ultimately it's exactly the same take-home message. What you can actually see is that when you do have an impact here in 0, that means that that's when the thermal safety margin has been reached, and everything above that value transforms into a negative impact, okay? So what this plot here shows is that actually, even though we have a higher increase in temperature at higher latitudes, what you do see is that these organisms, all right, so the dots are empirical data, these organisms, they are still falling in the positive zone of impact. That means they're still resilient to that increase. Now, when you go to near the equator, what you can see now is that the impact's negative. So that means that even though tropical regions are going to show a smaller increase in temperature, that small increase will have a higher impact in those communities than what happens in temperate regions, okay? And so the interesting thing about this is that applying understanding from thermobiology now inverts the predictions regarding the impact of climate change in ecological communities. That's one of the ways of seeing or trying to predict what might happen with climate change. And that's one of the ways that the information conveyed in the thermal performance curve can help us with these predictions. Let me show you another one. So there's another study that's now not talking about lethal effects, right? So we're not interested on the temperatures that might actually, drive a population extinct because it kills them, because temperatures surpass the critical limits, but instead we're talking about sublethal temperatures. So now, and if you look here at the plot on the right, what you do have is a predicted thermal increase, let's say in blue in cold environments and in red in warm environments, all right? And as you can see now, it's in the increasing part of the thermal performance curve. So we're not talking about lethal effects, we're talking about sublethal effects. Now there's another problem. As you guys can see in this panel, what you do have is that because the increase in this case, metabolic rate in energy expenditure is nonlinear, what you will see is that the same amount of increase in temperature, in this case, more or less 5 degrees in here at a cold temperature, will have a proportion, an absolute impact in terms of increase in energy expenditure that's substantially lower than what you might have at higher temperatures. So same delta T here, same temperature increase, all right? But the impact in metabolic rates in energy expenditure is substantially higher in warmer regions. And the reason for that is because you do have this nonlinear increase in performance as a function of temperature. Now, in the left panels, you see the consequences of this problem, all right? So you have here in panel A, essentially the change in temperature from 1980 to 2010 in different environments. You have the Arctic, North temperate regions, tropical and South temperate in different colors. And as we saw with the climatology predictions, you can see that the Northern hemisphere is more impacted. So there's an increase in temperature and higher latitudes. But the interesting thing now is that when we transform this impact in temperature into an impact in metabolic rates, what you do have is that the order now, once again, is switched, and you do have tropical systems having an increase now in what would be energy expenditure, metabolic rates, energy turnover as a whole ecosystem. And once again, the sublethal impact in tropical regions is going to be higher, or is predicted to be higher, than what happens in temperate regions. So even though, once again, you do have a higher delta T in temperate regions, you do have a higher impact, either lethal effects or sublethal effects in what would be tropical environments, right? That's very unfortunate. It's essentially where we have the more biodiversity, but that's what the data is telling us. Let's change a little bit now on the system. And let's talk not on difference on vulnerability to climate change across regions. Let's talk about terrestrial versus marine ecosystems, okay? So this is a brand new study, and you can see here some other interesting patterns that Pinksy et al. have unveiled. Essentially, you do have in terrestrial and marine ecosystems, this is the temperature, the highest hourly temperature that has been detected along a latitudinal cline, right? So essentially this is north, or sorry, this is south, and you have north here, and you do have present temperatures plotted for terrestrial ecosystems in green, and you have marine ecosystems in blue, okay? Now, with the projected warning, once again, we do have different scenarios. So what you can expect by the end of the 21st century is a higher increase in temperature for terrestrial systems. So there you go. I presume you're seeing the mouse in the screen. And you do have a lower increase in marine ecosystems in what would be the temperature. Now, once again, what happens when we study the thermal safety margins? And so these authors, they have compiled the thermal safety margins for a huge amount of different invertebrates and ectotherms. And what you can see are the results now in this plot. And essentially what you see is that even though terrestrial ecosystems are predicted to warm up more than marine ecosystems, right? And that's not entirely surprising. You do have air being substantially, has a higher thermal conductance than water. It has a lower specific heat. So you'd expect the air to warm up more if you want. But the problem now is that even though terrestrial ecosystems will be warmer, if you will, marine ecosystems will be apparently more impacted. And the reason for that, once again, is because CTmax, the maximum critical limits, are lower in marine organisms. And you can see this here, right? So essentially the thermal safety margins, there you are, they are somewhere around, let's say, 12 degrees, whereas, and particularly lower, once again, around the equator, whereas in terrestrial ecosystems, you do have now this smooth line showing that the thermal safety margins are higher in terrestrial ecosystems, okay? So once again, the predictions when it comes to thermal biology are inverted in comparison to what we would have just based on climatology predictions, or just predictions based on changes in temperature and nothing else. And just to recapitulate that we have this discrepancy between predictions not only in terrestrial versus marine environments, we also have it between temperate and tropical regions, right? And once again, this is at the global scale. One thing that I'd like to highlight here, just keep in mind, because I'm going to come back to this point again, is that the thermal safety margins in this case, look at these values, they're around 10 to 15 degrees Celsius. That would imply that these organisms, they could tolerate an increasing temperature of 10 to 15 degrees. I find that particular number quite strange, all right? And we're gonna be coming back to this value and to this observation soon. But to put things in perspective then, these large-scale studies employing the thermal performance curve, what they show is that you have the two big regions where you do have high diversity, right? So it's going to be environments that are warmer on a geographical scale, and tropical regions versus temperate regions. That's number one. And so tropical regions apparently are more threatened. They're ecosystems that are more vulnerable to global warming. And the second thing is that coastal regions are going to be also more threatened. And so keeping this in mind, let's check on the limitations of this the approach. And I would point out four that are interrelated, and essentially is that the thermal performance curve, it's a phenomenological curve. It's something that doesn't describe a mechanism, it describes a phenomenon, right? An increasing in performance and then a sudden drop. Many of the predictions regarding critical limits, they employ static endpoints. So they assume a single static value, right? CTmax equals, let's say, 30 degrees, and that's it. And for these reasons, they have a low resolution. So I'm showing this with this Europe plot here, right? So it's low resolution. And they also have a low predictive power. And what do I mean by that? The first thing, it's phenomenological, as I said, because we don't have much of an understanding of the mechanisms that underlie the curves that we see. So you can actually see in this case that at lower levels, you'd predict actually this asymmetric curve because you have a thermodynamic effect on enzyme rates of denaturation, or enzyme rates of reaction. And on the other hand, you do have a denaturation effect now that would affect these enzymes. And when you overlap these two curves, you get a thermal performance curve. Actually, these two, the performance curve shown in the bottom plot is actually the product of these two here. These are real numbers, all right? And the reference right there, there's a model that we can show that this happens. So you can get a thermal performance curve with no problems by including thermodynamics and denaturation. The problem now is that these mechanisms, they involve lower levels of organization, right? We're talking about biochemistry, we're talking about enzymes. But we also see, as I mentioned in the previous slide, that the curves are highly general. And many of the predictions that we have for global warming require that we know the impact of temperature in Darwinian fitness. And so that means that there's this huge question mark. How do these mechanisms at lower level translate into difference at higher level? All right, so one big question that I'd like everybody to keep in mind is, how does thermal adaptation emerge? If you have different populations and organisms showing different curves across different temperatures, how does that happen? And can we actually explain that? So that's one of the problems, it's phenomenological. The second one is that CTmax, which is, one particular point, one particular variable within the thermal performance curve that has been employed for predictions, is actually static, it's an endpoint, and it has low resolutions. Let me talk about this. First, many studies have shown that CTmax has very low heritability. Second, it shows reduced variation between populations and species. And third, many studies have shown that CTmax exhibits very limited latitudinal trends, essentially doesn't vary across organisms and across regions. So that has led many researchers to postulate that it has limited evolutionary potential. And we might be talking about a physiological limit that is absolutely general across organisms and lineages. Now, let me show you one particular result that I find quite striking from a work that we did in 2014. So I reanalyzed some of the CTmax data from previous studies. And when you plot just CTmax against latitude, you see certainly there's no association between latitude and CTmax. So apparently there's no trend. Now you see that the R-squared that this model explains is just horrendous. It's 2% of the variation. And the point with that is that we do have very low predictive power. Now, if you include the time, the exposure time that these animals have been exposed to during the trials, you see that the exposure time is very important. That shouldn't be surprising, right? If you're exposed to stress for a long time, you suffer more just because of time, more than temperature. And the R-square now increases to 54%. Now, the interesting thing is when you put the two variables in the same model, you unravel a latitudinal gradient. All right, and you also see the time effect. So apparently what has happened for a long time is that we have a low resolution, at least in part, because we consider the static point, when in fact thermal tolerance also varies in time, not only with temperature itself. And we can get to a predictive value now, or at least we can explain the variation in CTmax for something around 68% of the variance. And as you can see here, I'll just leave in the slide, the increase in CTmax with latitude, it's not small. We could be talking something about 10 degrees from the equator up to, let's say, polar region, not polar regions, maybe latitude about 60 degrees. And that leads us to a conclusion that instead of working with endpoints, we should be working with survival probabilities. So that should be our target variable, right? Something that's probabilistic, the probability to survive, the combination of a stress between temperature and the exposure time. And so suddenly you have now a switch, because we're not talking about a CTmax as a particular value. We do have the dependent variable becomes the probability to survive, to cope with the stress, and we have the combination of temperature and time. So when it comes, if you come back to the limitations, this is what we have. We have phenomenological framework that uses static endpoints with low resolution, with low predictive power. And essentially what we have right now in thermal biology is that we're gathering all this information in ecology, and we're trying to understand how this explains physiological variation. We get some predictions at the global scale, but that's definitely not enough good resolution. So we do have ecology explaining physiology, but we want to come back, and we're going now to future directions. We want to close the cycle and trying to employ physiological information to predict what happens to ecology. And with that, I mean, we have to transform a phenomenological model into a mechanistic one. We have to improve our understanding of mechanism. We have to switch from an endpoint to a more dynamic model and probabilistic one. And that will eventually increase the resolution of the models and the predictions, and of course, the predictive power of our model in thermal biology. So that implies that we need better frameworks, we need better theory to work with temporal and spatial variation to deal with extreme events. We know climate change is coming now, is increasing also thermal variability. This sort of framework will be very important to study what happens at the level of microhabitats and behavioral thermoregulation. Here you can see, for instance, this is a very recent study that shows that we can predict environmental variation and variation at the spatial scale at very, very minute scale. Here, each pixel is actually 1 meter square. So it means that we can now reconstruct temperatures across environments and microhabitats at huge, huge resolution. But unfortunately, our knowledge in thermobiology, what this might do to organisms, lags behind because of a lack of good models. And hopefully, better models would allow us to make better mechanistic niche models and better predictions in terms of the impact of climate change and global distribution. Having predictions at a lower scale would also allow us to start working with species interactions, so we can understand which species might be more threatened about changing temperatures in a particular environment. And that applies to invasive species, that applies to pest control, things like where do we want to plant our crops, depending on, some particular species that might be around and might threaten these crops. And of course, we might combine this sort of information, just as shown here, information on species that are more vulnerable to an increase in temperature with things like ecological networks and interactions. And we might eventually see how whole communities might be threatened by an imposing threat of higher temperatures. So just to summarize then, it's undoubtable that we do have thermobiology having progressed quite enormously during the past decades. It's been the progress has been tremendous. However, what we see right now, I've shown very, very recent literature, and many of the predictions and the level of resolution, we're still dealing at global scale, but we need better models and more refined predictions to bring this knowledge to a more local scale where conservation efforts and decision-making is taking place. And so just to close the presentation, I'd like to leave you guys with this quote by Michael Anguilera in his book of Thermal Adaptation, which essentially summarizes the main idea that I'm talking about here. And it's that the thermobiologists have described patterns and documented effects to the point that empirical knowledge has far outstripped theoretical understanding. So we need better models, we need better theoretical understanding of the problem. And with that, I will leave you, and thank you very much, and I'm open for questions.
Okay. So yeah, thank you very much for this talk. It was fascinating. Hold on one second. Thanks. Yes. So I'm just having a look at the questions here, and one which just come up from two people, Isabel Alonso and Louis Cortez. They're asking: can organisms migrate to avoid the CTmax? Is there any evidence of this?
Oh yes. Oh yes. So there are large-scale meta-analyses that are showing that animals are actually migrating. So what they're seeing, and many studies are showing that animals are drifting, they're moving towards the poles. Yes, and another possibility is to move to higher altitudes. So not only they're moving in latitudinal wise, they're also climbing, right? So they're going up mountains. And now the possibility to do that depends on the capability that the organisms have to do so. So some will have, higher dispersal abilities than others, and also the spatial scales that we might be talking about. So one interesting thing, for instance, is that altitudinal migration is actually way easier to do than latitudinal migration, because the delta in temperature by climbing, let's say, a few meters is substantially higher. You can drop temperatures incredibly faster than having to migrate many, many miles to get that same delta T, that same reduction in temperature that you would get. So, but the answer is yes, absolutely. Many animals can migrate. One question is, which are going to be able to do so?
Thanks. Yes, another question come in from an anonymous person. They ask, do any of these models account for other effects of climate change? For example, changes in precipitation or extreme weather events? I guess the extreme weather events are somehow accounted for in this CTmax approach.
Well, to some extent. I mean, I forgot to focus, to mention, essentially, because the title of the talk was temperature, I was very, let's say, unidimensional on the discussion of the subject, right? I didn't want to get right to global warming or climate change, including the other variables. But of course, better models require including things like precipitation. They require things like vegetation and primary productivity and so forth. So the single point that I was trying to make here is to emphasize temperature as one of the variables, and essentially how we focus on that one. But of course, it's undoubtable that the subject's way more complex than that, and more variables are included, and we should take them into consideration to get, you know, a better predictive ability.
Thank you. And we just have one final question from Ritu Yadav, who says, what about plants? So we concentrated on, I guess, vertebrates really here mostly. What about plants and other forms? Other people have asked about, you know, plankton and other types of organisms. Is this a vertebrate thing, or does this apply to all life?
It's a good question. This is a very good question. There's a problem here because we do have some limitations regarding the models that we can study. So working with animals, and in many instances with small animals, is just easier because you can quantify things like an endpoint. You know when they're dead. It's more difficult to estimate that in plants, and it's definitely, you know, you can do that with plankton. No doubt there is actually quite a lot of literature with plankton as well. With plants, what many people have done is estimated things at the population level. So essentially subjecting plants and seeds to different temperatures and seeing whether they're viable or not. But you cannot do this necessarily with organisms, right? It's very difficult to see exactly the moment or the particular temperature when a given plant was dead. I mean, you can definitely see it in the long term, but it's not something as immediate as, for instance, we do with animals. And that's why, to a large degree, the literature is biased towards animals, number one, and small ones, number two.
Okay, so I think just for time reasons, we're going to have to stop there. But thank you very much for this fascinating talk. Before we, and thanks again for joining us and for your time, I'm just going to say a little bit about what we're doing next week. Next week on Ecology Live, we have Kate Jones from UCL, and she's going to be talking about a topic which I'm sure will fascinate many people, how bats change the world. So I think we all know roughly what that's going to be about. Obviously, how land use change, and in particular, how land use change has influenced the exposure to infectious diseases, in particular, focus on bats in that talk. So I'm sure that will be of great interest to many people. So I hope to see you all again next week for the next installment of Ecology Live, and I'll leave you with the details of NHBS and their offer of free shipping for European orders. So thank you very much. Thanks again.